Okay, hello and welcome to the Nottingham Festival of Science and Curiosity 2021. Thank you for coming, whether you've attended or participated before, or whether it's your very first FOSAC event. It's great to have such a large virtual audience with us here today. We have plenty of events and activities for children, older students and adults to experience, including workshops, make-alongs, online talks and panel discussions. For further information and details, please visit our website, notsfosac.co.uk, or you can follow us on social media at notsfosac and share your thoughts using the hashtag CuriousNots or the hashtag FOSAC21. I'd like to introduce myself as the host for this evening. My name is Isabella Oakley. I'm on a placement year in between studying environmental science at the University of Nottingham. I'm really excited to bring to you What Does the Research Say? A series of online panel events discovering and exploring themes that influence STEM research around the world today and for the future. Also in the background, we have Rick Hall, who will be our tech backup and might be sending in some chats that you might see and monitoring the question and comment section. Today, we are bringing to you the future of medicine. The past 200 years have seen medicine advance at an extraordinary pace. People are now enjoying longer and healthier lives than their ancestors, but medicinal researchers want to take developments in health even further. With advances in gene editing, technological to overcome paralysis, and efforts to address high drug costs, the future of medicine is forever evolving and improving. In today's discussion, we'll be joined by Nottingham-based researchers as we explore the incredible new and potential developments in medicine and what they might mean for healthcare and society for the future. As a quick side note, before I introduce our panelists, we want to commit to create an open and safe environment where all attendees feel able to participate. Everyone involved with FOSAC's activities is asked to comply with the Code of Conduct. We'll be making our very own recording of the event, so we'd like to ask participants and auditors not to record, nor to take screenshots or photographs of the attendees and participants. Our team members will monitor comments and chat functions and take action where necessary. You can report an issue by approaching any member of the current staff team or send an email to info@notsfosac.co.uk. Now, with that out of the way, on your screens, you should be able to see our panelists, who I have the pleasure to introduce. I'd like to welcome to discussion our Nottingham-based researchers, Dr. Christina Lee, Associate Professor in Viking Studies in the School of English at the University of Nottingham. Christina will discuss her research with microbiologists, testing the efficacy of medieval remedies. Dr. Alvaro Mata, Chair in Biomedical Engineering and Biomaterials in the School of Pharmacy and the Department of Chemical and Environmental Engineering at the University of Nottingham. Alvaro will discuss research in tissue engineering and bio-inspired materials. And Nicola Richards, Senior Lecturer in Biomedical Science at Nottingham Trent University and has previously worked as a biomedical scientist for the National Health Service. Today, Nicola will discuss her research in haematology. Now, please feel welcome to contribute to the talks. This is very much an open discussion along with our panelists. You can leave your own comments in the chat function or questions in the Q&A section on your screen and throughout the event. We'll have an opportunity to discuss and answer these questions after all panelists have given their talk. I'm sure there'll be plenty to get through, so now's your chance to ask those thought about questions to an in-person researcher with plenty of knowledge and experience to share. So without further ado, I'd like to introduce our first panelist, Dr. Christina Lee.
So thank you so much for having me. And I just want to apologize because this is the future of the medicine and I'm going to take you into the past. And also you may kind of think, am I in the wrong meeting because there's someone from the School of English? Please bear with me. Next slide, please. I am a member of a group of people who are interested in looking at medieval remedies. So remedies that are mentioned in medieval manuscripts. And I'm a member of the Ancient Biotics group. Isabella, can you do the next slide, please? Sorry, I'm a bit like Chris Whitty here. Our Ancient Biotics group is a collaboration between people in microbiology, people who are doing computer science, and someone like me, who's doing the study of medieval manuscripts. And we got together by chance, and it was one of my microbiology colleagues who said, didn't your people have something against microbes? Looking at the problem that we're facing today, which is antimicrobial resistance, and something that will become a problem if we're not finding new antibiotics. And I was a bit skeptical, and I just said, well, you know, they did have remedies that seemed to be about killing bacteria, but there was a study before and it didn't work so well. And that made my science colleagues really curious. And so we decided, can we have the next slide, please? We decided to go to one of the remedies which may have been, you know, which were identified by a tech scholar as potentially efficacious, but which have been dismissed. And this is from a manuscript called Ball's Leech Book. A leech book is a medical book. It's nothing to do with the leeches. I know we're coming to hematology later on. So it's not about bloodletting. This is about medical plants. And this remedy, and you've got the original there, so you can see the manuscript, is against a stye in the eye. And we know that styes in the eye are caused by Staphylococcus aureus, one of the harmful bacteria that are also in their drug-resistant form responsible for MRSA. And it's very simple. It tells us it's a Beta-Lethodon, so the best of remedies. And what we decided is we're going to make this exactly as it says in the manuscript, exactly as it says in the text. So we have to consider that this time didn't have food processors, that this time had no fridges. And so we had to kind of think about how we're going to make this. We also tried to choose organic ingredients because we know plants have changed. We tried to find just the wine ingredient was really interesting because we had to try to find a vineyard that is in an area where we have vineyards in this time. We know where the manuscript is coming from. And so we did this. We made some batches of this remedy. It was interesting getting the email from my microbiology colleague saying it's self-sterilizing. Yeah, because plants also have bugs. They also have bacteria on them. And then, can we have the next slide, please? We tested it against Staphylococcus aureus, and I'm not sure if you can see the bottom one. We tried to do some tests with just simple ingredients or with a combination. This is a combination of various ingredients, and it killed 99.9%. So we thought we made a mistake. So we did this again, and we tested it again. We had one problem in this remedy, that one of the plants, Cropleach, we don't know what this is in modern English. We know it's an allium species, so we did 2 batches, one with leek and one with onion, because they are, I've been told, you know, from the chemistry, wide enough apart so that they may kind of have different effects. Both of them work a treat. So we did it again. It worked, you know, stuff again. It worked in all of our lab work. It worked, you know, stuff in vitro. We do know that a lot of remedies fail because what works in a petri dish doesn't necessarily work in life. So we send this off to a colleague in Texas who is doing mouse trials for potential new antibiotics. And we didn't tell the person what's in it. We just send it. And can we have the next slide, please? Our colleague actually said, oh yeah, we're getting so many of these, you know, yeah, I'm going to test them. What we found is that our remedy works better than vancomycin, which is one of the last defenses or one of the latest remedies. So what have we made? We are currently exploring, so we have tried out, you know, this remedy for safety. So because we needed to have a safety protocol, that this, because there's onion in it, and originally you were supposed to put this into your eye with a feather, not to be done at home. but this can be harmful. So we needed to make sure, and we have done, some of the research now, who have, sort of done some research on the safety protocol. We know that it is the combination of ingredients that works. It's not a single ingredient, because we got people saying, oh yeah, it's garlic. We know garlic works. But we also know that bacteria can evolve, and that a single ingredient cannot be as efficacious as this combination. So this still works. So as an upshot of this project, we are, still working on it. And at the end of it, I have given a list of literature if you want to read this up. There's a slide of, areas which you may want to read. We now in, can we have the next slide, please? We have now a new research project, and this is on nettles and networks, because we have realized that in quite a few of our medieval manuscripts, stinging nettles are part of the ingredients, and that they're often used to for serious wounds, so to cure wound infection. And so what we are doing at the moment is we have used networking theories, so mathematical theories, to get the most common combinations. And we were held up by COVID because the new nettles weren't out. We were out last year, but the labs were shut last year. And we are now going into the process of examining our findings, the findings that the medievalists have prepared in the lab, so the various combinations. So watch this space. I wish I could have given you a little bit of an outlook. Okay, so this is what I want to talk about today. It's a bit of a whistle-stop tour. I would like to do a thank you. This is on the slide, to our funders, which is the Royal Society. And I would like to say there is some literature if you are interested and want to know more. Thanks for listening.
Thank you, Christina. And I can see, I'll stop sharing my screen. The chat I can see has been popping up with lots of different things. And there's also been a question sent in. Can we ask this question now for you? And then if there's any more, we can ask at the end. So will the natural medication be produced in the UK anytime soon, as it is a better treatment? And that's from Eleanor.
Well, we hope so. There have been, sort of some, it was harder to get, sort of the funding, because there have been people who've been kind of saying, oh, well, this is not how you do history of medicine. Of course not. It's not history of medicine. This is working with scientists. This is truly interdisciplinary. But we are hoping that we will, sort of going forward very soon. Bear with us. That's all I can say. And don't try this at home. And I just want to say, I am not into homeopathy. This is into science. This is definitely underpinned by hard science, and it is something that can only be done by scientists.
Brilliant. Thank you, Christina. Just before we go on to the next speaker, I've seen some people commenting where they're from. We've got people from Boston, Massachusetts, from Nottinghamshire, and someone from Puerto Rico, which is brilliant. So hello, if you have just been joining, hopefully you caught on to the majority of Christina's talk. That's fascinating stuff. And now we're going to go to Alvaro for his talk.
Thank you, Isabella. Can you hear me okay? Yeah, okay, yeah. Great. So it's great to be part of this group, and it's international event. Okay, so I'll be speaking about tissue engineering and how materials are playing an increasingly important role. I will start taking advantage of Christina's venture into the past. I will start by a brief history of biomaterials, and we can go back to even 30,000 years ago. They have found materials being used for tissue engineering. What you see here, it's an eye prosthesis made out of wood and mud. So that's how we started. We started taking advantage of what nature would offer us in order to do an early type of tissue engineering. Then in the 1930s to 60s, there was an explosion of technologies. And the reason here, as you would guess, is because of the wars. And so, for example, a lot of materials emerged, materials for medicine, but they were not designed for medicine. They were designed for war. So, for example, if you look at this one, PMMA, this is a polymer that was used for windows of warplanes. And when the warplanes would be hit with bullets, the pilots would go to the hospital and the doctors would see the eyes of the pilots, which were fine, but they had pieces of glass in there, of these pieces of this polymer. And so that's how they found that this was a biocompatible polymer. And so this polymer later became, started to be used in contact lenses and other types of even implants. So that's how these materials were being used in medicine. They were designed for war at this time. Then from the 1960s, really we began to design materials for medicine. And so you started to have properties such as biocompatibility, really being compatible with biology, biodegradability, and a variety of different materials emerged that were designed specifically for medicine. And then this really takes us to now. And this has been, I would say, in the last 20 years, where we've been able to go down in scale. Nanotechnology became more readily available, the ability to work at the nano and the molecular scale. So we started to be able to understand how tissues and organs and biology works better. So we started to use building blocks that are present in biology. We started to be able to mimic biology, create materials that were intelligent and smart and responsive. And so in an interesting way, we went back to harnessing nature, to harnessing biology. Even going back 30,000 years ago, that's what we were doing. In an interesting way, this is what we're doing at the moment. Now we have the capability of understanding biology better and recreating it. And so when we're talking about engineering tissues and organs, really what we want to do is emulate what biology does. So let's briefly go into see how biology does this. What you see here, it's, hopefully I can play this. This is a video of a salamandra. This is a lizard that if you cut the arm of it, it will regrow the arm as it's showing in this time lapse. It will regrow the arm, the same length, the same fingers in the same location. It has a tremendous regenerative capacity. Now you can also have examples such as the dental enamel. This is a thin coating in our teeth that is the strongest tissue in our body. It lasts for decades. It has tremendous properties and it protects our teeth thanks to this organization of nanocrystals of calcium phosphate. So how biology is creating this and is being to generate these remarkable tissues? Well, it does really at the core by cells and how cells interact with their surrounding matrix or surrounding molecules. And that's what you see here at the top. It's a picture of a cell embedded in what is known the extracellular matrix. This is basically a fibrous cocktail of multiple types of molecules that are communicating with the cell and telling the cell to do what you're seeing on the left of the slide, right? Grow into different things, grow these remarkable materials. And so I like to compare this with the movie The Matrix at the bottom, because what we have at the top is basically a matrix that is full of information. It has a code, and that code is it's the language that biology uses to communicate cells, to communicate with cells, and really make these tissues grow and regenerate and so on. So what we want to do and what we want to do in the future is be able to understand this code, understand this language so that we can communicate with biology and hopefully work with biology to be able to recreate tissues and organs. And that's what we're doing. So before I tell you a few examples, and I show you a few examples, let me just briefly tell you what are these molecules that are making this happen. And to do that, we need to go and really to the core, to go down in scale and understand that biology and nature is hierarchical. That means we have atoms at the bottom. These atoms come together to form amino acids in this case, these amino acids would form proteins, and then proteins will come together and form cells and tissues and organs. You go higher in scale. Now, where you see proteins, this is an incredibly important molecule. And there are a few other molecules, such as DNA, sugar, and lipids, that are known as the building blocks of life. Now, this is, again, at the molecular scale, most of them are on a similar size scale. These are the ones that biology is using to build our bodies. And how does biology do it? How does it do it? How does nature do it? Well, it uses different processes. What I'm showing you here is the process of self-assembly. This is basically the assembly of disordered molecules. These are lipids, disordered molecules in water. What you see in gray are water molecules just moving around. And the green ones are multiple of these lipids that are just coming together by themselves, self-assembling into this lipid bilayer, into a higher order structure that creates a membrane. And these processes are actually happening in our body. So these are biological processes or tools that biology uses to regenerate itself, to create those structures that I was showing you before. And so what we want to do is use those building blocks I showed you before, use processes such as self-assembly, and be able to grow tissues and organs. And so I want to just give you a few examples of these are examples that we are developing in the lab, but there's many people around the world working on these and many other using different type of exciting technologies. This one here is looking at recreating this synthetic or this extracellular matrix. And what you see there, this yellow is basically the hand with the fingers of the cell grabbing onto this synthetic matrix. Again, trying to communicate, we're trying to communicate with the cell with this matrix. We are also developing materials based on these molecules, but can we grow them into more complex structures like these tubular structures? Can we create vasculature, for example, trying to grow vasculature? And what you see here is an example of a material where we are growing it by self-assembly. These are proteins that are assembling, similar to what you saw on those lipids, and they're assembling into this tubular structure. And what I want you to see here is how when it breaks, it heals up. So these are materials that also show self-healing properties, which is, again, one of the properties of our tissues. These are functional structures, so you can actually flow them. These are tubular structures that you can grow, and you can flow them and start having a functional vasculature. Let's go to a more robust material like dental enamel. Once you lost dental enamel, you grow it when you're a baby. Well, once you lose it, you don't have it anymore. We cannot regenerate it. It's actually acellular. Cells deposit it and then they die, and then you have that material. So it's really important to try to repair, to regenerate it. So we're working on different approaches to be able to organize nanocrystals of calcium phosphate and be able to grow into these structures. And then the last example I want to show you is a little bit of a different one, but again, it's one of the advantages of tissue engineering, and that we will see more and more in the future, which is, can we engineer disease? Can we engineer, for example, tumors, so that we can understand them better and we can discover better therapies and better drugs without the use of animals, without the use of people? So what you see here is, this is an ovarian cancer model. Here is a gel. This pink gel is the model, and inside of this gel you have these tumors that are growing inside, and we're using them to be able to discover new drugs in a safer and more effective way. So I've been showing you some technologies for really what is the future of tissue engineering. And so there's one last aspect, one last message I want to convey, which is, when we're talking about the future of tissue engineering, it's not only about technologies, but I haven't showed you really the human element here. These are the people that are making this happening. And so I don't know if you have this feeling, but I certainly do, is when we see, the emergence of technologies all the time, you tend to feel that the human element is put aside. And so I think it's really important, as we develop new technologies, that we always make sure we have the human element at the center of these technologies. So with that, that's my contribution. Thank you.
Thank you very much, Alvaro. If you'd be able to stop sharing your screen, I actually have a question for myself for you. And it's all to do with the regenerative structures. Does when they're regenerated, the different structures, does that affect their performance? Are they more likely to fail in terms of use and performance, or are they pretty much brand new or good as new?
So you're talking about the synthetic materials, or?
Yes, exactly.
Yeah, well, but that's one of the challenges. That's one of the challenges that you need to be able to regenerate to really to recreate what you had before, but perhaps you can even improve on what you had before.
Yeah.
So those are all options.
Excellent, and it's all part of research.
Yeah.
And to our final speaker, Nicola, if you'd like to share your screen, who'll be talking about hematology.
Right, hello everybody. I just want to thank you all for joining us today. You know, it's lovely that the festival has managed to pull together such a diverse group of scientists, and you know, it's wonderful to be presenting to such a diverse audience as well. So today I'm going to be talking to you about my field and the current developments in hematology. Okay, so to begin with, so I'm a little bit different to the other panelists in the sense that I'm a biomedical scientist, so I'm not employed in research, I'm employed in diagnostics. So I spend or have spent many of my days in clinical laboratories, as I say, diagnosing hematological disorders. So I'm a registered biomedical scientist in hematology and transfusion. I am also a senior lecturer in biomedical science for Nottingham Trent University. So what do biomedical scientists do? So as I said before, we perform advanced scientific techniques to help doctors diagnose disease and to monitor treatment. Okay, so what's hematology? Hematology is the study of disorders of the blood and blood forming organs. So as you can see in this picture, this is what I spend my days obsessing about, and that would be the blood cells. And biomedical scientists in hematology help to diagnose disorders such as anemia, sickle cell disease, haemophilia, leukemia. So depending on what part of the world you're from, some of these conditions may be quite familiar to you. They're quite common disorders. So to give you a bit of background, to start with how blood cells are made. So blood cells are made inside your bones, in the bone marrow. So if you can see on this diagram, the red areas of the skeleton are the areas where blood cells are actively being made during while you're an adult. And you can see that we have lots of different types of blood cells, and each of these different blood cells has a different important job to do in the body. So inside your bone marrow, there is a particular cell, a very important cell called the Hemopoetic stem cell. And this is quite similar to what Alvaro was saying, in the sense that the amazing thing about this particular cell is that it has the potential to become any of the other blood cells. And it's all down to how the matrix around the cell communicates and interacts with that cell to feed back messages about what the body needs in terms of the production of blood cells. So then there are various mechanisms in place in the body to make sure that this process is really strictly monitored and really strictly regulated, so that the body only produces exactly what it needs at any given time. But sometimes this tends to go wrong, because your bone marrow is a very, very active organ, and because it is producing cells at such a quick rate, the machinery inside the cell that helps regulate whether it's going to divide and how many cells it's going to make. Sometimes that can go wrong, and this leads to a condition called leukemia. So leukemia, again, it is quite a common disorder. It affects around 350,000 people worldwide every year. So, as I said, each time a blood cell divides, it has to copy the machinery that regulates its division. And because these cells divide a lot, there's increased risk that this machinery will be copied incorrectly. And if this happens, the cell division, there's a loss of control of that cellular proliferation, that cell division. So you end up with lots and lots of blood cells, and you think to yourself, well, you know, can you have too many blood cells? But the problem that you have is that these cells are functionally incompetent. So that means that even though you have lots of them, they can't do the job that they were meant to do. So these cells divide and they grow until they completely take over the bone marrow. And when the bone marrow is packed full of these, now what you would refer to as malignant cells, they're cancerous cells. When the bone marrow is packed full of these cells, it can't then produce enough normal cells. And as we've said before, these cells are really important in helping the body deliver oxygen to the tissues, in helping the body fight disease, in helping the body to form blood clots. So the body can't do any of those things effectively. So without the normal blood cells, if this condition goes on without treatment, it tends to be fatal. So, as I said, without treatment, it's fatal. So how do we treat it? So historically, treatment for leukemia was really quite aggressive. So the main options were to have a stem cell or bone marrow transplant, or chemotherapy. And as I said, these treatments are quite aggressive because chemotherapy basically works like a weed killer. So basically, chemotherapy is universally toxic. It's toxic to all of the cells of the body, but it will be absorbed quickly or more quickly by the cells that are growing and dividing the fastest. So that tends to be the tumor cells. But it will also include things like your hair follicles or the lining of your gut. For example, other healthy white cells. So even though it will kill the malignant cells, there's a lot of collateral damage. And similarly with bone marrow transplants, in order for the new bone marrow to be able to seed itself in the patient's bones, it's necessary to pretty much obliterate the patient's own bone marrow. So you can see that these treatments, they're very aggressive treatments, and there's a lot of side effects. However, it is an exciting time to be a haematologist, because for us, the future of medicine is all about personalised medicine. So as I say, historically, you would take, you know, a member of the public who needed treatment, and then there would be established treatments, and you would try an established treatment and see if it worked, and it would work on some, but then there would be others who it didn't work on. So for the ones who it didn't work on, then you'd need to try a second line therapy, which again would work on some, but then there'd be others that they didn't respond. And then similarly, if they didn't respond, you'd have to try a third line therapy, and so on. And all the time that you're having to try different therapies, you're losing time, you're giving the tumour time to grow and do damage. And similarly, you're also exposing these patients to really aggressive treatments. So what can we do? So a lot of the research in haematology is about being able to identify specific biomarkers in our different patients that tell us which therapy the patients will respond to. So instead of trying all of the therapies on all of the patients, we're able to test the patients, and more specifically, we're able to test the cancerous blood cells to see what these blood cells are expressing. And that helps us to identify which particular therapy they're more likely to respond to. And so obviously that saves a lot of time for the patient. Now, the other thing that we can do, which is really good news for the patient, is we're doing a lot of research to kind of work out specifically where the machinery that helps regulate cellular division has gone wrong in the different types of leukemia. Because if we can work out specifically where the machinery has gone wrong, we can actually target the therapy specifically to that point that's gone wrong. Which means instead of this broad, aggressive treatment, we can say, well, it's like instead of having a generalized weed killer, you're able to go individually and just pick the weeds. So an example of this kind of approach is a drug called imatinib. So this is a drug that's used in chronic myeloid leukemia, and scientists have been able to identify exactly the point where the cell's replication machinery has malfunctioned, and this has allowed them to produce a synthetic antibody that specifically targets that malfunction. So research is being carried out now at pace, really, to identify other potential targets in the different leukemias, because what we found with imatinib is that this has been highly successful in treating patients. It has a lot less side effects. There's much better chances of the patients going into remission. So this is kind of like the driving strategy now for developing new drugs and new treatment regimes for haematology patients. So that was my view of the future of medicine. Thank you very much for listening.
Thank you, Nicola. Some fascinating stuff. We've had a question from Paul, if you wouldn't mind answering. Great to hear Nicola talking about personalised medicines. This seems to be a great area for treatments and advancement. But what are the major challenges in terms of safety and efficacy and production?
Very good question. So major challenges in terms of safety, as with all things, I think that this is where the time is spent, really, because I think once you have a mechanism that you can test, you're able to formulate kind of potential drugs quite quickly. Where the time is spent is on the trials, and kind of seeing, you know, the drugs progress through like phase 1, phase 2, phase 3 trials. In terms of efficacy, I think what we're seeing is that these treatments tend to be much more effective once they've passed through the clinical trials. These drugs tend to be much more effective than conventional medicines because they're so well targeted. And sorry, what was the third one?
Production was the third one.
Production, yes. So this is another kind of major hurdle in the sense that when these drugs first come out on the scene, they do tend to be eye-wateringly expensive. So yeah, that's the big hurdle at the minute, is kind of being able to manufacture these drugs at a cost-effective rate. Because obviously, as the NHS takes on more of these drugs, as more of these drugs are cleared for use by NICE, it is really kind of looking into, can we justify the expense? But that said, you know, as technology improves for the production of monoclonal antibodies, because a lot of these are based around monoclonal antibodies, as technology improves in that area, the cost should come down.
Thank you, Nicola.
Isabella, can I jump in, since I think all of us spoke, and I think there's going to be a discussion, but can I jump in to follow on Nicola's on that?
Yes, of course you can.
Question.
Nicola, sorry, do you mind stop sharing your screen?
Yeah.
Thank you, and we can bring us all together.
Yeah.
Excellent. Off you go, Alvaro.
Yeah, no, just I mean, just to follow up on Nicola's point, especially the last point she made on the financial part, because we're also aim to work, and as Nicola said, this is at a very exciting time to work in different type of therapies because of the possibility of personalized medicine. But the idea of personalized medicine moves away from the idea of scalability, you know, in a way. So a major challenge, I think, in general, is how we manage to combine the necessary scalability to maintain financial viability with the fight with the personalized element, the unique personal element. And I'm just mentioning because there are many therapies, even in tissue engineering, that we are working on with the aim of personalizing them, and that's a challenge because of the costs of really focusing on one patient, and knowing that currently medicine and therapies are, you know, we are in a system that depends on scale. You can have a great medicine, a great drug, but if it's not scalable, it's going to be very difficult to develop. Yeah.
Christina, do you want to jump in? Were you going to say?
I just wanted to jump in because I've really enjoyed, you know, both presentations. But Nicola, it just made me think, you know, in my medical text we have alternatives for remedies, sometimes up to 64 alternatives for a specific disease, or, you know, where there's different combinations. We also see something in its infancy that is, we might call gender-based medicine, because we have different ways, you know, so this for men and women in some aspects. And I just, you know, for us, it has been very difficult to kind of decide which one to concentrate and whether they were actually meant to treat the same condition, or meant, you know, stuff as something, this works here, but that one doesn't work there. So I'm really fascinated by the personalized medicine.
Yeah, I mean, I think it's a very good point that you make about like differences in gender. We also see differences in efficacy across different ethnicities. We also see that there's differences in terms of kind of who is more prone to particular disorders. And I think what has been difficult in the past is the fact that a lot of medicines, when they're developed kind of on what may be quite a homogenous group, and then that's kind of scaled up. And then it's only later that you see that actually there are discrepancies on how, you know, efficacious that is between different groups. So yeah, it's very interesting.
Thank you, Nicola. We've got quite a few questions and comments, so it's great to have this discussion, and please feel free to jump in and ask each other. So coming back to you, Christina, there's a question that asks, is there any truth that metals are a cure for rheumatism?
I have heard that before. It's not in my texts. Can I just say, kind of, you know, so wiggle out of that one, please?
Okay, I've got another question for you. So I remember learning about leeches as a medieval remedy back at school. Is there medicinal remedies that we still use today, or have they mostly been adapted and replaced?
Yes, I mean, we're not using the leeches anymore, and even in my period, people are very, they seem to not be too keen on bloodletting, because they find lots and lots of excuses on why you shouldn't let blood. So doctors kind of have all these kind of, obviously there's some kind of patient choice. Obviously, essentially all modern medicine at some point is based on a plant-based derivative. So aspirin, you know, I have birch bark, you know, still in some of my remedies, and aspirin, it was discovered, you know, from birch bark. So, you know, we do still use some of the ingredients, but, you know, developed and refined, and obviously we do know now exactly how they work, or mostly how they work, and that's different from that period.
Great, thank you. Do you want to jump in, Nicola?
Sorry, yeah, I was just going to say it's really interesting, because what was fascinating about Christina's talk was how these remedies kind of, we discover the science behind them and then they kind of come back into favour. So, you know, we are actually seeing new uses for leeches. So for example, one of the things where leeches are being used are, for example, if you have, say, a severed finger, they can use the leech to put on the end of the severed finger to, the leech's saliva has an anticoagulant in it, so it stops the blood from clotting and gives the surgeons a bit of time basically to be able to reattach lost digits and so on, because that blood supply is being maintained.
I think that's so fascinating, because there is what is described as ethnopharmacy. So I think, you know, stuff there's, it's not just here, you know, with medieval remedies, but I know that the Quave Lab, for example, in America uses, they work with Native American remedies. I do know, you know, so that in Brazil and in Africa and in China, you know, so there's people working with this. And it's aside from sometimes people have this idea that, you know, these things are just kind of secondary, but there are scientists going back and just saying, okay, you know, we may have some efficacious plants or some animal, you know, we shouldn't forget animal medicine, and we may kind of think about it. I like that also where Alvaro was kind of coming in. These are the natural components, and it is, it's not, I mean, you know, so what you're doing, Nicola, my medieval people would say it's a miracle. You know, this idea that you can cure something that is so invidious, but kind of coming back, you know, sort of to the basis of these natural stepping stones and put them back together in a different order.
Thank you, Christina. Anything from you, Nicola? You. Thank you. Okay, Alvaro, coming to you, we've got a question. What's the process of regenerating tissues? They said that they are aware, you mentioned atoms and proteins and how important they are, but how do you go about doing it?
Well, that's it's a big question. I mean, it's really understanding that process is something that people are still doing. That salamandra that I showed, there are people, many people trying to understand all the minutia that is taking place to try to develop therapies for, say, for humans. I would say in a simple answer, I would go to how we stimulate cells. So cells are a fundamental part of who we are. I mean, we should think of ourselves not as an individual, like you see one person. It's really billions of multiple organisms that remarkably are coordinating into you. But it's so really, to regenerate, we need to work at that level. We need to work with cells. We need to stimulate them. I presented how the matrix does it. And there are ways in which we can now engineer materials and other systems to talk to cells, to stimulate them to grow and so on. But there's still a lot to be learned, and you know, that still needs to be understood. There's a lot of room to. We've come a long way, but there's a lot of room to move forward.
Of course. Thank you, Alvaro. Back to you, Nicola. We have another question. Can genetic makeup be a factor in affecting personal medicine, linked to the mention of gender and ethnicity?
Yes, is the short answer. I think it's interesting because I think what we're certainly seeing is for different conditions will affect different ethnicities, for example. So one of the fields that I'm particularly passionate about is hemoglobinopathy. So these are conditions like sickle cell and thalassemia. So we see quite a lot of geographical variation in terms of which communities are most affected by these. But we're also seeing now that we're kind of looking at the data more deeply, that we see discrepancies in a lot of different disorders. So for example, prostate cancer, for example, being something that we're now seeing a lot of research tell us that it's a lot more prevalent in males of African origin, a lot less prevalent in males of Southeast Asian origin. So yeah, there's definitely ties that we're seeing, and a lot of research going into kind of where are these differences and, you know, why do they occur?
Isabella, can I jump in again?
Yeah,
Follow up, Nicola.
Go for
Sorry,
it.
Nicola, I have a question for you on that, because I wanted to ask you about the epigenetics. So a long time ago, I think people used to think that genetics was the whole thing. Then there was this wave of epigenetics, you know, how our genes may be modified by our environment. What's the current, you know, school of thought in this in leukemia, let's say, or in
that it's a complex puzzle in the sense that if your genes are, say, your genetic potential, and then kind of how your environment interacts with that to manifest kind of your being, so to speak, it's quite complicated to kind of then claw through the data and say, well, what is specifically down to the gene, what is specifically down to cultural or lifestyle factors, because there's a lot, you know, those issues tend to confound each other quite a lot. So for example, if you look at diabetes, you know, we know, for example, that there's a genetic component to diabetes. However, when we look at studies, we also see that you are more likely to become diabetic if you live with a diabetic. So, so you can see that there's a genetic component, but the fact that someone who is not genetically related to you is still more likely to become diabetic because they live with you kind of highlights what the lifestyle components are as well. So it's quite a difficult puzzle to pick apart.
Interesting. Thank you.
Yeah.
Thank you both. Now, before we wrap up, because I'm conscious there's lots of discussion going on, which is fantastic. Nicola, there's a question for you. What can we learn from animals like the naked roll mat that are allegedly immune to cancer? Have you had an experience with that?
I have not, but there are, I think, you know, there's certainly, I believe, a lot of research going on to kind of, like, as Alvaro was saying, looking at these amazing properties that the natural world shows us and kind of saying, can we replicate that?
Brilliant. Okay, so I'm going to go around each of you now. If you'd be able to just give a quick summary and maybe something that you're excited for in the field of your research, or a new advancement or development that's going to be happening in your field. So to you, Christina, if you'd be able to wrap up.
I'm extremely excited that materials which have been used in the past may make a difference in the future, and that we can harvest or data mine, you know, sort of these manuscripts for potential solutions to problems that we've got now. And mentioning Nicola, diabetes, they didn't know about diabetes then as well.
Thank you, Christina. Yeah, all about building off what we've known from the past. And to you, Alvaro, if you could wrap up with a summary of your talk and what you're looking forward to for the future.
Yeah, I think I'll stay with the same theme where I focus, which is the idea of learning from biology, which ties with what both Nicola and Christina have been saying, learning from biology and trying to, you know, not only get inspired by it, but can we cooperate with it in order to, you know, to develop therapies and technologies.
Thank you, Alvaro. And finally, Nicola, if you'd be able to summarise.
I think I'm excited about the explosion of new discoveries that we're finding in terms of really unpicking the mechanics of these different tumours and being able to, like, have a whole host now of, like, potential targets for therapies.
Excellent. Thank you ever so much. So as we've come to the end of our discussion, I'd like to thank everyone for listening and contributing with your questions and comments. A virtual round of applause to Christina, Alvaro and Nicola for their valued time and sharing their knowledge and research with us today. We hope you've been inspired and your curiosity captured. If you have any feedback or comments, you're welcome to email us at info@fosac.co.uk. We have 2 more panel discussions exploring sustainability and diversity in STEM, so please head over to our website and sign up for a place if you're interested. We hope you enjoyed the rest of your FOSAC 2021. Have a wonderful evening and we hope to see you soon. Thank you very much.